This is going to be a very fast overview of the highlights of the DNA lab. I have my electrophoresis chamber there. I've gotten down an Erlenmeyer flask. I'm going to add 150 mLs of TBE buffer to that. Then, after I fill that up, I'll pour it into my chamber. Now it's time to work with the samples of DNA. I brought down 5 microtubes and I've labeled each one according to the table in your lab. Now it's time to fill the tubes with some DNA. So I bring down the DNA ladder, which is going to be the one we compare everything to. I put 0.25 mLs in there. And I'll do the same for all the rest of them. So then the mother's DNA, I bring that down, add 0.25, and go ahead and fill out the rest of those tubules. Now it's time for the 2X gel loading buffer, and I'm going to put 250 microliters in each one. It's time to draw up 5 microliters from each of the samples. I'm doing the ladder first, which is our main one that we look at to compare things to. I drew it up and now I take it over and put it in lane 1. And you'll see in your table that's what it tells you to do. Then I go to the next one, the mother's. Once again, 5 microliters. And I put that in lane 2. So I go ahead and do all of them, dispersing them in the correct lane. Now I'm ready to process it. Unfortunately, it was a bit off the screen. I put the voltage at 100, and then down on the left is this timer. So as soon as I hit on, I go down to the bottom left corner and turn my timer on for 1 minute. That's very important. And then you turn it off when 1 minute is passed. If you go under or over, it'll change your data. And so here's my data. And it says to start at the bottom that first one is at 50. So I enter that measurement at 50 base pairs. Then I go to the next one, 76 millimeters, and that is my 100 base pair number. And so go through all of those and transfer those numbers across. That's what you're going to use to compare your mother, child, father possibility 1 and father possibility 2. Here's the mother's, and if you look at that top one, that was 1,000. That was the next one down was at 900, and the next was 800. So that was the mother's reading right there at 837 millimeters. So you go through and figure that out for all of them. And then when, of course, they have the same thing, that's what we're talking about them being related, because they have the same base pair sequences. All right, hope that helps. Email your instructor with any questions, and good luck.